This Week in Virology, the podcast about viruses, the kind that make you sick. Hello, ECV! Well, that was pretty bad. Let's try it again, because you don't want other places to be louder than you, do you? Hello, ECV! Yeah, that's much better. From Microbe TV, this is TWiV, This Week in Virology, a special episode recorded on April 29th, 2019. I'm Vincent and you are listening to the podcast all about viruses. Today we are coming to you from Rotterdam in the Netherlands. We are at the meeting of the European Congress of Virology for 2019, and that is a meeting put on by the European Society for Virology with participants from over 50 different countries, and some of them are out here in the audience today. And we're going to do two TWiVs, unprecedented at any meeting, and we have taken for each one a selection of some of the diverse virology that we have here in Europe. And joining me today to help me co-host this first TWiV, this is her second time on TWiV, from Erasmus Medical Center, Marion Koopmans.
Hello there.
Welcome to TWiV, welcome back. And you're a co-organizer of this meeting, right?
I am.
And was it a lot of work?
It is, but we have a good team. We have a great local club.
Right.
We had a couple of TCs, and then the rest was a breeze, actually.
And tomorrow, TWiV will have Ben Burkout, your other co-organizers, to be my co-host as well. All right, so Marion will help me speak with our three guests who are here in front of you. And on my right, from the Bernhard Nocht Institute for Tropical Medicine, Stephan Gunther. Welcome to TWiV.
Yeah. Hello.
Morning. I bet you're wondering what this is all about, right?
Actually, a bit, yes.
Well, we're just going to talk about science, and we happen to be recording it at the same time, and millions of people will listen. Well, not millions, but many thousands of people will listen. Next to Martin, from the Friedrich Loeffler Institute, Martin Beer.
Hello.
Welcome to TWiV.
Thank you.
And finally, from Wageningen University, Vera Ros.
Yes.
Is it good, Ros?
Ros, yes.
Okay, welcome to TWiV.
Thank you.
And you're the youngest of the group here, right?
I think so.
And we have a great audience, so thanks for coming. And by the way, we have some TWiV T-shirts to throw out later, okay? So this is going to keep you here. So this is a special edition T-shirt. It's the TWiV World Tour 2018 T-shirt. All right, the front is a cool design by Sharon Isern. She made this design of a virologist walking into a virus particle with headphones. And then on the other side is a list of all the places we went, including 3 places in Europe. We were in the Netherlands, we were in Belgium, and we were in Switzerland. And so I'm going to throw these out at the end, so stick around for that, because these are pretty expensive shirts. So, and if you don't want them, give them to someone else. You don't want it?
Yay!
That's just one. I have 4 more here, and if you don't get one today, come tomorrow. We have 4 more. All right. So I first want to hear from our 3 guests just a little bit about. If you want a T-shirt, you can have one too. I'll send it to you. I want to hear a little bit about your training, where you were brought up and raised and educated. Let's start with you, Stephan.
Yeah. Yeah. Actually, I'm an MD by training. I started medical school at the Charité in Berlin, and I have to confess I was never really sure if it is good if I treat patients. So already during my studies, I got an interest in virology. Actually, I don't know why. Maybe because these viruses, they are so small but make so much damage with just 4 genes, 3 genes. So it's I mean, yeah, it's amazing. It's not good, but it's really interesting. So I got this interest into virology, and I actually had 1 year off during my studies and did research in Munich. And my former teacher, Hans Will, he worked on hepatitis B. He went to Hamburg, had a professorship, and then he invited me to come to Hamburg. Actually, this was also the time of the unification in Germany, so positions in the clinics have been extremely rare. So maybe this was another aspect. So I had no chance to get a clinical position. So I said, okay, let's continue working in the virology field. And then I worked really 5 years hardcore on the desk, pipetting, sleeping overnight in the lab on hepatitis B. But already at this time, I gained an interest in working on more with patient materials. So we're looking at hepatitis B variants, which develop in patients, and to better understand why these variants develop, what they do, what is the molecular biology of these variants. And so often in the career, a lot of things are by chance. At this time, Germany had a law which meant that you had to leave an academic institution after 5 years. Otherwise, they had to give you a permanent position. So, which of course means you had to leave. So they didn't give you the permanent position. So I had to leave. And at this time, I already stayed in Hamburg. And at this time, a position was offered at the Tropical Institute. And I remember, so I applied, I presented all my stuff on hepatitis B, and they said, yes, it's okay, but we don't want you to work on hepatitis B, we want you to work on Lassa. So I said, hey, what is Lassa? The hell, I never heard about this virus in my studies. Yeah, so I looked it up, and so, oh, it's similar to LCMV. So because LCMV, we had a group working on lymphocytic choriomeningitis virus, which is the pet of the immunologists. And so then this was in '98. So and then I started. I said, okay, fine, if you want me. So I go into Lassa, and we really started from the scratch at this institute and developed this line of research for Lassa fever at the institute. And I always kept these two aspects. So still working really much on the basic aspect of the disease. So we have structural biology group, we have immunology groups looking at these aspects of the disease, but always keeping an eye on the patients. And now we're developing and looking at diagnostics. We're now involved in clinic or planning for clinical trials. And so this actually what I like, and actually the Institute also giving us the opportunity to have this breadth working really right from patient service, from diagnostics up to basic science.
So in the end, you do touch patients.
I would say I touch patient samples, which is often also crucial for the clinician. That someone tells them, oh, this is disease X, Y, Z.
Right. Got it. Okay. Very good. Martin, how about you? Tell us about your background.
Yeah, I'm a vet. So I studied veterinary medicine in Munich. Mm-hmm. So, and my goal was to be a cow vet.
A cow vet?
So go out, cattle, be a cattle doctor.
What do cows drink?
Water.
Okay.
So it's still my favorite animal. But I did an internship in virology in Munich, and that was nice. So then I did my PhD on an important cattle virus, on BVDV, bovine viral diarrhea virus, and did my PhD on it, did my habilitation in Munich. So I stayed in Bavaria, very much my favorite area at that time. And then, as it was said, after some years you should change. So I got the offer to go to the island of Reims, so to the Friedrich Loeffler Institute. So I would say the birthplace of virology. So the area where the first animal virus was described, and I took over the reference lab for another a cattle virus, the bovine herpes virus type 1, because there's a big eradication program in Germany. And after some years, I think it worked well. We are now free from this disease in Germany, so everything is fine. I promised we will be free until my retirement, but this is 17 years ahead. So from that point of view, we did well. We will see how it works with BVDV. And then I got head of the Institute of Diagnostic Virology. So that's really still the focus on diagnostics. And from that point on, I would say from 2006 on, it was out of my hands because we had one outbreak after another in Germany. So blue tongue disease, H5N1, Schmallenberg virus, and we could work on that. And at that time, we were the first to use next generation sequencing, which is now one of the favorite methods we use for metagenomics. So, and I talked today about the Borna virus story, which was found by metagenomics. So I think it was the last 15 years, I would say, everything happened by accident. So it was not really planned, but I'm happy with that.
That's what Stephan said, things happen by accident. So when you were in vet school, is that when you thought that you might want to do science, or was it even before?
No, it was in vet school mainly. Before I was interested, but I thought it's best to be outside and work with animals. So that was really the goal, and I still like to work with cattle, for example.
So this morning in your talk, you showed a picture of an island that you work on, right? So where is this?
This is in the northeast of Germany, in the Baltic Sea area. So when I told my wife, we will move from the middle of Munich to Greifswald. That was not so easy.
Sure.
So I always said other people go to the US or to China, so I went to Greifswald. So but we decided we will not move away. And the travel time is the same, yeah.
So you live on the island?
No, I live close by the island, so 12 kilometers away.
Oh, you take a ferry?
No, there's
a bridge?
On the picture you could see there's a driveway since the 70s.
It reminds me of Plum Island, right off Long Island, except there's no land bridge. You have to take a ferry.
In former times they took a boat and then they had a cable car to go to the island. And you can swim if you want. So that's okay. So it's I think it's a nice area, but you can now go by car, so it's pretty easy.
Is there any wildlife on the island?
Yes, we have a sentinel station for influenza, so we have mallards there, and we have a camera there, and so we know what there is. So there's stone martens, there's badgers, there's wild boar, there's deer, but they are all outside the fence.
Right. Well, at Plum Island, they don't want any animals, but apparently deer swim across the water, and they can go up to their neck and swim, and they get on the island. They don't particularly like that.
I think it's not a problem. It's good to be on an island. I think Friedrich Löffler made a good decision because they had the outbreaks with foot-and-mouth disease in the city of Greifswald, and now we have all the filter systems and whatever. So
Vera, tell us a little bit about your.
Yes, so I'm trained as a biologist. I actually did my studies in Wageningen, where I'm now. And I've always had a keen interest in host-parasite interactions, and I'm mainly focusing on microorganisms and interactions with arthropods, including insects. I did my PhD in Amsterdam, at University of Amsterdam, and I focused on Wolbachia, which is now a famous bacteria for having antiviral properties, but at that time that was not discovered yet. And it's also highly interesting because it manipulates the reproductive system of insects. So it can make species completely asexual or turning males into females. And I've especially investigated this asexuality in mites, so not insects, but arthropods. And more from an evolutionary perspective. And after that, I've worked for a postdoc at the University of Pennsylvania on Lyme disease, so Borrelia bacteria, mainly focusing on the antigenic system of these bacteria, so how they evade actually the immune system of the host, so they have a surface protein that can change fairly quickly. And well, after that, I moved into virology. So I'm not trained as a virologist, but moved into virology during my postdoc. And now I'm deeper and deeper into virology. So I work on viruses and insects. And I started as a postdoc, and I'm now in a tenure-track position as an assistant professor in virology in Wageningen, and I study how, one of my interests is to see how viruses can actually change the behavior of insects. So this is a very nice example of parasitic manipulation. There are more parasites that can do that. So also fungi, bacteria are known, and they're very appealing examples of crickets, for example, committing suicide by jumping into water when they are infected by worms. And I work at these caterpillars. They are showing zombie-like behavior. So once they get infected with the virus, they start becoming hypermobile. So they move around over larger distances, and they also climb to elevated positions in plants, and that's where they die eventually. And they actually start climbing when they're really ill. So you can see they are getting pale, they're really heavily infected, and they still manage to climb. So the virus is inducing this climbing behavior in these caterpillars, and we really want to know how these viruses can do that. So yeah, I think we have a very neat system at hand because we can genetically modify these viruses in the lab pretty easily. And then we have these caterpillar rearings, so we can study how they show this climbing behavior.
When did you get interested in science? How long ago can you remember?
Well, actually, when I started my studies, I was interested in doing nature conservation. But at that time, I discovered there was much more behind this. And when I initiated my study in biology, I became fascinated by discovering how nature functions, how it works. And I was really getting more excited during my studies, and I knew I wanted to do a PhD and continue in science. Yeah.
So if you want to hear about Marion's background, you can listen to TWiV 413, which we recorded in Boston. You remember that, right?
Yes, I do.
That's why I'm here, because we did a TWiV and she thought that was pretty cool. And you said, when I organize a meeting, I'll invite you back. So here we are. And that TWiV was called Partnerships, Not Parachutes. And that's something that, Stephan, I'm going to talk to you about, the idea that you want to help a country. You don't parachute in and take things and leave, but you actually establish partnerships. And that was the theme of that TWiV. So Stephan, let's talk a little bit about your work. When Marion suggested I invite you to this, she said you're one of the few people who have a presence on the ground in Africa. What does she mean by that?
Yeah, I mean, the actual presence, I counted, I stayed meanwhile something like 1.5 years in Nigeria, cumulatively, not at a whole, cumulatively, over the past 10 years.
Right.
Yeah, at some point during our research, actually, we felt that, of course, you have to look at the patient and see what is happening in human. Animal models is okay, the basic research is okay, but if you don't know what is really happening with the patient, I mean, you won't move forward. And eventually, we don't know. So Lassa is called a hemorrhagic fever. For those who don't know it, it's a bit like Ebola. It's not as severe as Ebola. It's not as contagious as Ebola. But still, we have a case fatality rate in the hospital of 30% or even more. So, and of course, we don't have a clue why patients are dying. So what's really happening? We still, actually, I have to confess, we still don't have a clue really what's happening in a Lassa patient. Everything is something like a host response apparently against the virus, and a completely dysregulated host response. So 10 years ago, we have to learn from patients, and we established a collaboration with an institution in Nigeria, in the south of Nigeria, in Edo State, where there was some evidence that Lassa is endemic in this area. Very few publications. Actually, there was one site in Sierra Leone, but there were others working in Sierra Leone. We said, okay, let's look at new sites in West Africa. And this has developed as a very, very fruitful collaboration. Of course, I have to mention the name of this institution. It's called Vera Ros Specialist Teaching Hospital. This is like an academic hospital which is linked to a local university under the federal government. And they really showed a very, very strong interest in working on Lassa. And at this time, you have to imagine no one in the country was able to diagnose Lassa. So sometimes samples were sent out of the country, then sometimes they were diagnosed in South Africa, some went to CDC. But Lassa is not, you cannot distinguish Lassa from other febrile diseases in an African setting. So like from malaria. So maybe in the very, very end stage of the terminal disease, you might have some signs which might lead you to the clinical diagnosis. So the first point we said, and we came around the country, and it was actually very funny. So we met them the first time. We were introduced by a professor with whom we worked for a long time in Lagos, and he introduced, and they explained us. We had a board meeting, and I mean, it was my first time I had a board meeting in Nigeria. It's very fair because it opens with a prayer, it closes with a prayer, and then we had this meeting with this, and they said, yes, we want you to have, we want diagnostics here. If you want to do research, we want diagnostics. Well, we said, okay, let's try this. I mean, we never did this in the past, setting up a diagnosis. We said, well, we need to set up PCR diagnostics, because usually everyone was looking for ELISA techniques. No, ELISA won't help you. So you need PCR. So, and then some notes were taken, and in the evening, they brought us to a big reception. I said, why we are here? So we said, okay, because you are here. So all the consultants, everyone was in the room, and they started. But we said, okay, but first, please sign here. So, and they came with the first MOU saying, yes, we are going to train staff, and we set up this lab. And this was in 2007. It was really the start of a great journey. We have a lot of friends now there. So they set up the lab, we came in with diagnostic equipment, and we maintained the diagnostic now for 10 years. So meanwhile, they are going to test something between 1,000 and 2,000 samples per year, meanwhile from all over the country. It helped us to develop new assays. So maybe I can spend a few words later on this, develop new diagnostics. And of course, this was crucial for patient management, because now they could set up a ward for Lassa patients, and once someone was tested positive, so the positivity rate was just something between 5 and 10%. So this gives you an impression that, of course, it is not that you clinically can have a clear suspect. So 5 to 10%, they could be treated on the ward. We discovered that renal failure apparently is involved in the pathogenesis of Lassa, as it also is in Ebola. So they set up dialysis. Apparently, this is also in supportive care, improves the outcome of Lassa. So, and this was the last 10 years journey. Of course, with all the capacity building we did, we have now set up with funding from German government, what we call it Training and Clinical Trial Center, which is a 500 square meter, very nice building, the nicest building in Edo State, actually, with lab capacity for GCLP, with offices for the various projects, also with a very nice accommodation for us, with a terrace to the bush. Yeah. So and this is the point where we are, and we're now in a stage where we can start with clinical trials for drugs, for vaccines. And fortunately, after Ebola, there has been a change in the funding policy. Now, WHO and big funders look also very much more onto these diseases, which are listed on the so-called, WHO blueprint for urgent research and development.
So Stephan, can I ask you, because there's a lot of discussion now about the need for research capacity building, so that the research agendas are really tuned to African research priorities. Yeah. How is that working for you? So you just talk about diagnostics, but that's of course a different step. Did that work well? What did it take?
Meanwhile, a lot of research, in particular clinical research, can be done already in country. For example, we have fax machines are on site. Essentially, the samples which we move out of country, are mainly meant for virus isolation in the BSL-4 lab, so that also the virus is conserved. That's quite an important aspect because we need also isolates for validation of diagnostics. But a lot of research can be done in country, and for example, the last 2 years we moved into the country with so-called Marion sequencing devices, very small. Still, it is that this research is driven by foreign partners. So this is not that we can say that the African partners can drive this research. This would be the next step, clearly. But of course, the first step is that we see what is feasible, of which type of research is feasible under resource poor conditions. So then the next step is that then we train people on site and then implement this type for long-term, research technologies on site. This is a long journey. For the sequencing, for example, we discovered that it is no problem for our African partners to do the pipetting. Yeah, but the challenge is the bioinformatics. Yeah, even for the Marion, which does not generate that much data, but this is a challenge. So we are looking at pipelines which can be locally implemented. We need bioinformaticians locally, and it's not only the transfer of the technology. The key point actually is the human resources. Yeah? So over the years, we trained so many researchers, lab staff. Often then they went to other places, so it's a continuing process. But what you need is also a change in the educational system in the university systems in Africa. So they have to produce eventually the human resources for the research. And the leaders in research. We always talk about the ownership of research by the African partners. You can take ownership only if you have the qualified human resources. And we are on this track. And others too, of course, but I think it will take some time because you need a lot of elements. Yeah.
So, how many cases a year of Lassa fever are there roughly?
They diagnose, I think, the last two, what we call outbreaks. Actually, Lassa is an endemic disease, so we have it all over the year. It is 99% of the infections are direct transmissions from the rodent to the human. So this is different epidemiology. Like Ebola, you have one introduction and then the explosion in humans, human-to-human transmission. Here, everything is still reservoir to humans. And this is more or less happening all over the time. But we see peaks of disease January, February during the dry season. We see and then the diagnosed cases in hospital is something about 200, 300, depending on the height of this peak. Definitely, there are much more Lassa virus infections. This is just one center in Nigeria. So now the other centers in Nigeria need to be developed with diagnostics, with clinical capacity, and also to treat patients. So I would guess you'll get up to maybe at least 1,000 clinical infections. And then we know that Lassa, in most cases, Lassa is a mild disease. So sometimes...
So there's been some increase in Lassa in the region. And I have been... So we talked also a little bit about, can you find common patterns? There's also been an increase in monkeypox cases, where the question is which rodent types could be involved in bringing that to the people. And in my mind, I've been wondering if there's some connection between the two. I mean, is there something in general changing in favor of rodent populations in the region that could be looked at, thinking about ways to advance our knowledge for early detection?
There are partners in Africa working in the field and looking at the rodents. So it's one, apparently the primary reservoir is one rodent species called Mastomys natalensis, but they recently found also other species which probably are a reservoir, like Mastomys erythroleucus. So it means the reservoir could be much wider than we initially assumed. Then we also have to confess that we know that Lassa has several lineages, but not for all lineages it is really clear that this is the rodent reservoir. Maybe there are other reservoirs for specific lineages. Yes, we have some evidence for this. So that means that the ecology of the virus is might be much broader and much more complex than we initially thought. What actually causes the increase of the cases is not well known. Climate factors are apparently one, because this is very much correlated with the dry season. Once the rain starts, they know 3 weeks after the rain starts, the Lassa cases go dramatically down. But we know there are fluctuations which are over annual, so not within a year, and this is very, very poorly understood. We don't know if this has something to do with the rodent dynamics. It might be better diagnostics, of course, a bit like diagnostic epidemic might also play a role. Yeah, more awareness might play a role. So, but it's very difficult to distinguish these aspects.
So, given the 1,000, you said maybe 1,000 or more cases, right? Why is it that Ebola gets all the attention in the press and hardly ever Lassa is mentioned?
Oh, in Nigeria, it is well represented in the press.
I'm sure, but say in the U.S., which is my experience, no one ever writes about Lassa, yet there are always many cases, but Ebola always gets written. That's a hard question. You may not know the answer, but I'm just curious of your thoughts about that.
I don't know. I mean, of course, we had this large Ebola West African outbreak, a magnitude we would never expect that it could happen in this magnitude, and it threatened also the Western countries. So we know that maybe the difference is Lassa is not as contagious. So we know this. Lassa, there are documented cases, I think in the literature, something like 30 to 40 documented cases of export of Lassa from the endemic area to Western countries, US, Europe, Japan. And as far as I remember, there's only one proven secondary infection, and this was 3 years ago in Germany, where we had a expatriate case of Lassa, and the undertaker then who prepared the body for the burial, he became infected. So this is the only really proven secondary infection. So all the other Lassa cases had close contact. So it is not as contagious as Ebola, and maybe this reduces a bit the fear.
Maybe, but I would suspect that most people don't even know that. Yeah. So I don't know how that would play into it. Right?
But nevertheless, there is the public health impact in Africa is
very big. Besides Nigeria, what other countries are impacted?
Oh, it's Nigeria. We know now about Benin Republic. In the north of Côte d'Ivoire, Mali is affected. And then a huge endemic area is Sierra Leone, Liberia, and so the Man River countries, Sierra Leone, Liberia, and Guinea.
Okay.
So speaking to that contagion issue, the first hospital where a lot of the Ebola patients were in West Africa was a specialized Lassa fever clinic, and despite their level of knowledge. Yeah. A lot of the staff there, the medical staff, got infected with Ebola. So, because of the higher transmissibility of Ebola.
Right. Let me ask you one more question, then we'll move on to Martin. So, you said every infection is basically a new spillover from a rodent reservoir. Yeah. So that is, if you take one home which has a case, it will have its own virus, and another home is a distinct, or are they? Is it the same virus circulating in rodents that can infect multiple times?
I mean, this is more or less inference both from field epidemiology. Yeah. So that there is no direct link between the cases, but it's also the molecular epidemiology, because what we and others did over the years is a full-length sequencing, as I mentioned. So we did it with the Marion. And you can a bit like calculate how many exchanges you might have according to the molecular clock of the virus, how many exchanges you might have by human-to-human transmission. So this is in the range, of course, this is like a distribution, 1, 2, 3 exchanges. But most of, if you do the sequencing of the Lassa cases, so the differences are much larger between the strains. And this is also what we know from the rodent population. So the strains are diverse. So it means even the infection in this house compared to this house, yeah, it's diverse. Yeah. So, and if we have human-to-human transmission chains, then usually it's just one exchange, 0, 1, maximum 2 exchanges.
Yeah.
So,
right. So you said there are lineages, 6 lineages or something like that?
Yes, I think we are now at 6 or 7.
So are there any?
It's not formalized, so we.
Yeah, there may be more.
Gunther lineages.
And are there differences in the disease caused by these lineages, or transmission, or anything that we can tell?
There are essentially only 2 lineages where we have sufficient information. This is lineage 2 in the south of Nigeria, and this is lineage 4 circulating in Sierra Leone. And but this is more a guess, because there are no really hard data, systematically collected data. I would say it is comparable. But maybe there are small differences, but for this we would really need systematic studies on patients with the same protocol, with the same diagnostics at the 2 sites.
Mm-hmm. And finally, as far as we can tell, Africa is the only site where you find Lassa virus, or you can find it elsewhere?
I mean, if you bring the rodents here to the Netherlands, and they can spread here, so you can also have Lassa here.
You could, but
no one has tried so far as I know.
Let's move on.
So, the Mastomys natalensis, and as far as I know, also Erythroleucus is found only in sub-Saharan Africa.
Okay, and that limits
so what is a bit surprising is that Lassa is confined to West Africa, even though the reservoir is found across Africa. We find other viruses, apparently not as pathogenic viruses, in Mastomys in other parts of Africa. So maybe it can still spread a bit in sub-Saharan Africa.
Okay. All right. So I don't think we've ever talked about Lassa much on TWiV, but I read a book called Fever in 1970, which is about the first Lassa outbreak in Nigeria, by John Fuller. And at the time I was a college student, and that's what made me want to be a virologist. I thought that was the coolest story. You know, the story involved Columbia Presbyterian Hospital in New York City, because Penny Pinio, the first nurse, among the first nurses, she was airlifted back to Columbia. She recovered. They kept her serum, and they used it to treat Jordi Casals, who infected himself in the laboratory at Yale. And I thought this was an amazing story, so it's always had a special place. Now, Martin, back in 2015 on TWiV number 347, we talked about your paper on the variegated squirrel breeders and them getting Borna virus. And so I heard you talk this morning about this series of transplant recipients. Summarize that story for us, the New England Journal paper.
So the first paper was about these squirrel-transmitted Borna viruses. So for us, this was really a big surprise that these squirrels were Borna virus positive. It was a new one, and we could really relate it to the cases in the breeders. For me, everything was new. Also, that there is people in Germany breeding exotic squirrels from Asia and everywhere. So we had a lot of telephone discussions with them.
Yeah.
So it's also a very special type of people, so which breed that. So it was interesting in many ways, I have to say. But they were, when we could tell them that we don't have to kill the animals to test them, then the compliance was pretty good, and we think we have most of the animals in Germany tested. So we provided a program that they could send in feces samples and swabs.
So these are variegated.
Variegated and so-called beautiful squirrels. This is the Asian type. So Calosciurus is its zoological name, and we still don't know, was it introduced from Asia by importation, or was it introduced from Central America? And we know that one of the persons who has introduced these animals to Germany in the mid of the 90s also died with the same symptoms. So we count this person as a probable case of variegated squirrel infection. And the sequences look like that it was a single introduction. And at the moment, we even cannot exclude that there is any other small mammal which has transmitted it to the squirrels. But I think this is, the probability is much lower for this hypothesis. And we were sure that this is the zoonotic one and the classical Borna is the non-zoonotic one. But it turned out that this was wrong. And the first case is the people sent us from all three transplant people, recipients, the different clinics decided to send us sample more or less in the same week independently, because they have seen that we did some metagenomics on encephalitis cases. And therefore we received the samples. One was also for rabies exclusion, because the FLI has the WHO reference lab for rabies exclusion. And we did in parallel our pan-Borna virus PCR and NGS. And the next morning, everything was clear that in these transplant patients, we find a lot of sequences of classical Borna virus, that it's more or less identical for the two kidney recipients. And we had to rethink this story. So we wrote it, we submitted it, and at the same time, the group in Erlangen, Armin Enser and his colleagues had done NGS on an encephalitis case of a young man who died also of acute encephalitis without any transplantation. And this is the two papers coming up, and the story is now different again. So we have two mammalian Borna viruses, which obviously can be transmitted by spillover. And the reservoir is, and there it's a little bit like the Lassa fever, that you have one reservoir. This is the shrews, this bee-colored white-toothed shrews. And from there, it's obviously transmitted, and you have these different clusters. And we can see from these clusters that this is always a single transmission event. And the dead-end host, now the humans are also dead-end hosts, cannot transmit it further. So it's really restricted to the central nervous system.
Right. So the transplant, the person who donated the organs was presumably infected, but you weren't able to test those tissues.
We could not test the tissues, so that was not able. We had some sera, and they are positive. They are weak positive. They have around 1 to 100, so that it indicates infection, but there was no indication of Borna-related clinical signs. So we don't know, is that, has he donated in the incubation time, or is it a kind of latent infection? So both kidneys were infected. We had material from the kidneys because they are tested pre-transplantation, and we received material before transplantation, and this is negative. And then they made an explantation, and this is highly positive. So this really developed after the transplantation, but it was transmitted by the donor, and the liver donor survived with some severe lesions and blindness and so on. So, which is also disease reported by horses by surviving Borna. So I think the new story is that this Borna virus is also the classical one. They can be zoonotic, and the humans, like many other animals, are accidental dead-end hosts.
Right. So do we have any history of rodent contact for the donor? He didn't keep squirrels, right? He didn't.
No, he didn't keep squirrels, and the only thing we also have there is he's a cat owner.
Cat owner, okay.
So I don't know if this really plays a role, but cats are catching shrews. Yeah. And the shrews can be infected. They don't like to eat it, so they bring it home, and there has to be some shrew-human contact. And this is, I think, for the future, is something we really have to find out how this happens. And we even don't know the transmission route. Is it intranasally? Do I need scratches or whatever?
Yeah. Well, you can imagine he might have picked up the shrew from the cat and got virus on his hands. Yeah. But the rodents excrete it, right?
The rodents are persistently infected. They are healthy, from what we can say, how a healthy shrew looks like. And they run around and look nice. So in Gießen, they had infected shrews in a holding, and they look normal. And in some areas, up to 30% of the shrews can be positive. And they are full of virus, also in the central nervous system, but also in the kidneys, in the skin, in the urine. So there is virus excretion.
And they're fine.
and they are fine.
As far as you can tell from a shrew, right? Yeah, that's great. So are these in shrews or squirrels anywhere else in the world besides Germany?
So this Crocidura leucodon, the shrews, they are in many parts in Europe. So, but the Borna endemic region is smaller. So this is really German-speaking countries like Germany, Switzerland, Austria, and Liechtenstein. So we don't understand why this is the case. The variegated squirrel Borna virus, we still don't know. So we are looking in samples from Asia and Central America. And there is a Bodivis-2 virus found in Austria in one sample. So we don't know if that is in further small mammals. So I think it's now the time for metagenomics, looking by NGS, looking with a Borna virus real-time PCR in many shrews worldwide and see if there is, or squirrels, if there's further Borna viruses.
So can I ask, because the older Borna disease story suggested that humans also could get behavioral problems, like psychosocial problems.
Sadness.
That was then discarded with the with the realization that the diagnostic workup wasn't correct. But now you're getting backtracked on that story. So should we be looking again at the behavioral effect? And I was also struck by the potential parallel that we have here.
I was afraid about this question because it's a very difficult one. But at the moment, I really think we have to separate it. There's the old story. For sure, there's no scientific where you can say a scientific reason. There you can say all these tests were correct. No, everything we know is it's not like it was thought, and other groups showed that it was wrong. That was really lab contaminations and some problems. So, and also if we look at these old samples, we cannot confirm these things. If we have now the story that the people really get sick, they die, and it's most of them. So, and it looks like it is in the horses and in all other animals. There might be a phase in this when they get sick, that they have also these changes, but a few weeks later they are dead. So at the moment I would say we should stick at this very clear picture, which only now stares with, if you look now at the data, it seems very logical. And you cannot understand that 20 years ago the people were not testing encephalitis cases. So it would have been possible. There was a nice antibody which could detect it. So if you have looked at cases like it is in horses, in humans, it would have been found more than 20 years ago. And I think we were really on the wrong track.
But something that has struck me in your work is also the power of metagenomics, but you managed to fish out the right sequences. So maybe in this case, they were so abundant that was evident, but in the Schmallenberg, I remember the first story, the first samples, there were 3 reads or something like that were Schmallenberg. What told you to look for those as the real culprits?
So I
think it's still art, but
it's all starting with the sample. If you have not the right sample, you can do what you want. And for Schmallenberg, I have to say, many thanks to the technician again. There were 15 samples, and he picked up 3, and that was the 3 positive ones. And we had only 27,000 reads, and 7 were positive for Schmallenberg. So that was, there was a lot of sequences in it. For the Borna virus, for the squirrel Borna virus, we took all samples of the squirrel, and that was less than 5 reads of hundreds of thousands of reads. And one reason is the system we use is a metagenomics software we called REAMS, which is a workflow where we really test against everything in GenBank, and we even look at every single read which might be interesting. It's only at first information for us, and the second step is always interesting reads. You have your sequence, you order a real-time PCR and confirm it. And that's the next step. For Schmallenberg, we even could isolate the virus and could go back into the animal, which is, I think, an important step, because normally you have now a lot of sequences where nobody knows what does it mean? The virus is not existing. There is no Henle-Kochsche postulates.
Yeah.
That's very often missing. And I think for Smallenberg, we could show the full picture. For the Borna virus, we have now the first isolates from the human cases, and isolates went into the monkey model. So I think this is very complete. And but it always could be that's one read in your metagenomics, which is the hit. And the problem is if that is far away from what you have in the databases. And today there was a discussion, makes it sense to have all this sequencing done with all these viruses nobody needs? And I would say, yes, it's pretty important because this makes the database analysis for future outbreaks much easier. Because the next virus could be some from an insect, which everybody said it's useless, but it's a related one of the next zoonotic one. And then our metagenomic software is easily detecting it, much faster, much easier, and we will not miss it. Therefore, upload everything in good quality, please. Because that's also a problem that we have a lot of sequences in low quality, and this low quality sequences slow down the process. So we need high quality sequences, but every virus you can get rid of, because this could be the basis for good analysis in the future.
So is it too early to say we should check organs for Borna virus here in Germany anyway?
I think it makes no sense. This is so low incidence, and we have no chance to check it before, because also the organ sample we had from the kidney was negative in the PCR. So this might be a low level of replication. And even the serology is difficult because we don't know if it makes sense. So the recommendation together with the people from the transplant centers was not to do any testing because there is a so low risk and not a good chance to get it by testing. So
what about asking about animals in the household? It's not your first question you think of if someone is dying, but
I think the risk is it's so many people having that, and at the end, I think we have to say that the absolute numbers are so low.
Yeah, sure.
That I think that's a fortunate thing. That it's something which is, if you are affected, it's really unfortunate, but it's seldom.
Well, if you have encephalitis or some CNS symptoms after a transplant, then maybe then we should have antivirals ready, but we don't, right? There's no, or maybe there's a broad spectrum RNA antiviral that could be used. So that's argument for developing such.
It's an argument, and I would also say it's an argument if you have unknown diseases which are severe, we should use more and more metagenomics because there might be other viruses out. For me, it's an example, and it's also an example for if we import exotic animals, it could transport Lassa fever, it could monkeypox come in, it could come in Borna viruses, so all these exotic animals, and I think it's tens of thousands of reptiles and small mammals coming into the EU, and I think we have to be prepared, and we use it as an example. That we say, Borna viruses, you need, if we have this contact and people having contact to exotic animals and getting diseased, we should use metagenomics as a standard technology.
But of course, as you say, for the squirrels, if you don't have to kill them, it's better, right?
It's much better.
You can swab them.
It was also, as I said, it was 20 squirrels and one was positive, and the official vet catched this one. So if he would have caught the other one, I think it would still be unsolved.
Right. It's luck.
Yeah.
You need luck. Good fortune. All right. That's a great story. Thank you, Martin. All right, Vera, let's talk about what you do. But first, could an insect virus infect people? You think one day?
Well, there's a distinction, of course, between insect-specific viruses and viruses transmitted by insects. So there are many examples of where the insect is the vector. That's always the question when I lecture about using viruses for biocontrol purposes. So like baculoviruses that I study are used widely for biocontrol, and their creating awareness is also very important to know that they are safe. So I would say there is no chance that they can infect humans. And even for baculoviruses, they're even used in protein expression systems and also in gene therapy. So they're very safe in that way, and there's no way they can transfer to infecting humans, so they're very safe. But also, a lot of the large-scale sequencing projects, they now show that there are so many insect viruses. We only know a very small part of the insect viruses that are present. So more and more insect-specific viruses are being discovered that we know nothing about.
Yeah.
And I think that is a big challenge now to get more knowledge on that. And it also appears that there are some insect viruses that cause these acute infections, but now it also appears there is a huge number of viruses that are causing covered infections, so persistent or latent infections, which might again affect also arbovirus transmission, but also other insect virus infections. So there's a huge knowledge gap there, and a lot more to be known.
So I always tell my students that if you eat cabbage, you get a lot of baculoviruses,
yes,
right? And they pass through you, right?
Yeah, we have all eaten it. So they are present on crops a lot, so, and they're also used in biocontrol and very safe in that way. Baculoviruses are very sensitive to the alkalic midgut of insects, so the alkalic midgut of caterpillars, so the pH is very high, and that's why they disintegrate. And then so they have a protein matrix protecting the actual virus particles, and that dissolves in the high pH environment, and then the virus particles can bind and fuse to the microphyllae of the midgut cells and enter the midgut cells. But they can happily survive a passage through a bird gut, for example. So birds are also active spreaders of these viruses over long distances. And humans also can, they survive a passage through the human gut system as well. Yeah.
All right, so you mentioned at the beginning you're interested in how viruses manipulate host behavior. And you mentioned caterpillars crawling up high in trees, and then the birds eat them, right, and spread it. So in the lab, give us an example of how you study this.
So, we infect the caterpillars with these viruses, and we look at their behavior. So we have a big arena in the lab where we have a video camera that we can look at their behavior in the horizontal plane to see how much they move.
Another TWiV location.
Yeah, yes. That's right.
And but we also look at climbing behavior, and at the moment we use marmalade jars.
Marmalade.
I know in the US there's also a lab studying it. They use Coke bottles. That we use to study this climbing behavior. We have also done this in the greenhouse on real plants, but that has a lot of challenging aspects, and the outcomes so far are the same. So this marmalade jars help us to facilitate the setting. We can record their position every day and see how they climb.
Right.
And we found that these caterpillars in our system are attracted to light. So we can provide light from different directions, and we noticed that they are positively phototactic, so they are attracted by light once they are infected with the virus, and they go up to the light.
But not when they're uninfected.
When they are uninfected, they show behavior going up and down and up and down, but they do that in the dark as well as in the light.
Okay.
So there is no difference between light and dark. And the funny thing is that they climb when it's dark. So the climbing occurs during the night prior to death. So they don't need the light when they climb, and we have identified a time frame prior to climbing where they really need the light. And somehow they remember or they respond at a later time point to the direction of this light, which is, I think, a very intriguing phenomenon. So they're kind of primed, or maybe some pathway that is affected by the virus is triggered at that point, making them climb at a later time point to where the light is coming from.
Mm-hmm. Are you interested in understanding how infection does that?
Yes, we are. We're actually also, we started off by looking into the genes of the virus that are involved in this behavioral manipulation. So, and we've focused so far on two genes. One is a phosphatase, and that is linked to this hyperactive behavior. So if we remove the viral gene from the genome of the virus, they don't show this hyperactive behavior anymore. And the other is an EGT gene. It's an active steroid UDP glycosyl transferase. It's an enzyme that interferes with the molting hormones of the insect. So a lot of insects, they have different stages in between which they molt, and this enzyme is blocking molting. And it's thought to be beneficial for the virus because they don't molt. When they molt, they usually stop eating. So if they don't molt, they continue eating and grow bigger, so more virus is produced. And first it was identified that this EGT was causing the treat-up disease, but now it becomes more clear that actually a lot of these caterpillars have very specific behavioral patterns related to molting or eating. Some species go up to molt, some go down to molt. Some show daily movement patterns related to feeding. And I think it's more this interference of this EGT gene of the virus with these behavioral patterns that we see, whether they go up or down and whether they die at elevated positions. And these viruses seem to have found ways to do that at the right time point.
Is this so that the caterpillar gets eaten? Is that the idea and transmits infection?
The idea is that the caterpillars are at high position, so and they die at an elevated position, so they actually become a sack full of virus particles.
Oh, right.
So they liquefy, they melt, so to say, they become totally fluid. The idea is that the virus is spread because it's at an elevated position over larger distances, either by rain or by wind. But also they're more easily picked up by birds, so they're transmitted at longer distances. Yeah.
So do you want to also know what these viral gene products do to the caterpillar? Is that?
Yes, that's our next step. We're trying to identify what the substrate of this phosphatase is, and then to look further into the pathway leading to these changes. And one of the interesting things is now that we're also looking into the brains. So caterpillars also have brains. They're far less complex than human brains, but they have a lot of conserved pathways that we also see in humans. So we can actually learn a lot about studying insect brains on how human brain functions. And we're now looking into whether these viruses manipulate this central nervous system of the insects to achieve this modified behavior, which is very intuitive to think because behavior is regulated by the brain. So it might be either from the outside, but we have now found evidence that the virus actually go into the brain at a few days post-infection. So we have made fluorescently labeled viruses, so we can easily track these viruses, and we see that they enter the brain at, in our case, 2 days post-infection, and then they also show specific localization in the brain. So they don't go everywhere, they go to specific cells, to specific neurons that they infect. And it shows a very specific pattern. The downside is that we hardly know anything about insect brains. So especially, most of what we know is from Drosophila. From caterpillars, there was nothing known. So what we started with is to create a model of the brains of caterpillars. So that's a few of my students worked on that, and we now have a nice 3D model of a brain of a caterpillar. And that will help us to identify the areas and the cells that these viruses go to see whether, for example, they interfere with the production of neurotransmitters that are often involved in regulating behavior. So we try to see whether production of serotonin and dopamine is changing when they are infected with the virus. And we can then use the wild-type viruses and compare to these mutant viruses that do not induce this behavior. And that makes a very nice comparison because they both get infected, but there is a difference there.
So these viruses kill the caterpillar.
Yes, they do. Yeah.
And you suggested that maybe there are other insect viruses that cause persistent or chronic infection.
Yes.
And maybe they get into the brain and manipulate behavior, right?
Yes, indeed. There's not much known on that part. There was recently a paper on a plant virus, actually, that causes a persistent virus in its vector, and it can manipulate the behavior of the vectors. Also, plant viruses can manipulate behavior of the vectors. And actually, the interesting thing is that these baculoviruses that we study also appear to be causing covered infections. And it's something else that I'm looking into at the moment. So if you screen caterpillars from the fields, you will almost surely find baculoviruses at a very low level infection. So they're horizontally transmitted, cause acute infection, but they're also vertically transmitted, causing these latent or persistent infection. And that's also, and whether they manipulate behavior, we don't know. So that's something to look into.
It's great.
Yeah.
Do you think
this is really what we like to bring together? This, so very different worlds, but it's so thought-provoking.
Sure.
You know, what you say, we can learn about the functioning of the human brain from the caterpillar brain. Yeah. You know, think of that. Think of the Borna questions. I think it's very fascinating.
Do you think human viruses manipulate human behavior?
Who knows? I mean, we've seen that. Isn't it with, for instance, dengue infection in mosquitoes also changes their smell preferences, things like that. So,
well, even if you have a flu, you start sneezing, which is in fact physiological, but kind of behavioral change as well to enhance the transmission of the virus. So I think there are many ways in which the viruses aim to increase their transmission, including also behavioral changes. Yeah. And of course, it's very difficult to experimentally study that.
Sure. But if people have hemorrhagic fever or encephalitis, their behavior is changed, right? But I'm wondering of more subtle things. Maybe you get smarter if you have a certain.
Well, there are some studies that claim that toxoplasma, so which is not a virus, but that it's affecting the driving ability of people.
That's right.
But I think there's also under discussion whether that is really true or not. But and also in the early 20th century there was this big flu outbreak. Mm-hmm. And after that, there were a lot of neurological diseases like schizophrenia and other, and there's also suspicion whether there is a link between this huge outbreak and these later neurological diseases. But also that is very hard to demonstrate whether that's the case.
Yeah, in big populations, it's hard to show that it's this and not something else, right?
Right.
Well, we're running out of time, so let me start to wrap up here. I want to ask all of you one more question. Very quickly, Vera, if you had not been a scientist, what would you have done?
If I wouldn't have...
If you had not become a scientist, what would you have done?
I don't know.
No idea.
Become a nature conservationist.
Okay, that's good. Yeah. Martin, how about you?
Farmer.
A farmer. Oh. Would you have cows?
Yes.
Of course. Stephan, what about you?
I think when I was a young boy, maybe a pilot.
A pilot?
Yeah.
An airplane?
Yeah.
Yeah, okay. Yeah.
Right.
Cool.
I have a flight simulator.
You do? Okay. Nice, very good. What about you, Marion? What would you have done?
I think I would have gone into acting.
Acting?
Yes. Okay. I did that as a hobby for many years, but then it was hard to combine it with being a vet. So
I think to be a scientist, you have to act somewhat sometimes, right? All right, one more. I can't resist. RNA or DNA?
Oh, this is very hard for me.
Yeah, I'm sure.
No. I like viruses with 4 genes.
So you... that means RNA. That must mean RNA, right?
No, hepatitis also has 4.
Okay, so you're not going to say RNA or DNA. No. How about you, Martin? RNA or DNA?
RNA.
RNA, Vera?
DNA.
DNA?
RNA.
Wow, cool. All right. So that is our first of 2 TWiVs here at European Congress of Virology. You can find TWiV on any podcast player. Please subscribe so you get every episode, and we know how many people are listening. We love getting your questions and comments. TWiV at microbe.tv. And if you really like what we do, consider supporting us financially, microbe.tv/contribute. My guest today from Erasmus Medical Center, Marion Koopmans. Thank you for joining us. From Friedrich Loeffler Institute, Martin Beer. Thank you so much. Thank you. And from the Bernard Nocht Institute for Tropical Medicine, Stephan Gunther. Thank you for joining us. And from Wageningen University, Vera Ros. Thank you so much for joining us. I want to thank the European Congress of Virology 2019 for bringing TWiV here, in particular Marion. Thank you so much for inviting him. And I want to thank ASV and ASM for their support and Ronald Jenkins for his music. And so now let me give you some T-shirts here. Does anybody really want one? All right, here's one really big.
You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.